Hello?
Hello?
Yes, we have more 4 minutes to start the program, okay?
Okay.
So, the thing is okay, is by control. So, in 3 minutes more, I start the program.
Okay.
Thank you.
Paolo?
Yeah.
I'm going to start by just talking for a bit, and then after a bit, I'll pull up the first slide. So if it doesn't work and you can't see it or something, please say something. Step in so I can fix it, okay?
Okay.
Because I can't tell from here if it appears or not.
Okay.
Are you at the University of Sao Paulo?
I work in Institute of Technology from São Paulo States, not in University of São Paulo.
I see.
But the institute is inside the university.
I had a visitor from the University of Sao Paulo who's interested in nano safety. There's a Guilherme Lenci Silva.
Yes, I know him.
You know him?
Yeah.
Right.
We work in many occasions in seminar, etc. He's a very good person, a very good scientist.
Yeah. You spent some time here a year or two ago.
Also, I have a friend here in your university. Is Timons. I think you know him.
What's the full name?
Timons Robert. John Timons Robert.
Oh, right. No, yes, he wrote me. I know, I'm sorry, yes, Timmons. Yeah, he wrote me about this. And he told me that he knew you, and I don't know him well, but I've met him several times. He's a global climate change person.
Yes. He worked so much with this climate change.
Right, I knew that you were connected. I'd just forgotten.
1 minute more.
Okay. Now, what happens if does this have to run for 1 hour, or what happens if it's 45 minutes?
It's okay.
Okay.
Okay, there is no problem. When you want to finish, there is no problem.
Okay. I don't know how long. I'll just talk, and you can ask questions if you want, but I can't quite guess how long it might be.
Okay.
It won't be over an hour.
Bom dia, boa tarde, boa noite aos internautas que nos acompanham neste programa Nanotecnologia do Avesso, hoje no seu número 461. Fazemos este programa dedicado ao público não especialista, porque é este público que paga as pesquisas sobre nanotecnologia realizadas no Brasil. Por fim, fazemos este programa porque acreditamos que ser livre é ser bem informado e a divulgação científica contribui e colabora para o aprofundamento da democracia e da cidadania no Brasil. Hoje, conforme tínhamos anunciado, estamos conectados com o professor Robert Hur, diretamente da Brown University, da cidade de Providence, nos Estados Unidos. Eu vou passar a palavra para ele, ele vai se apresentar e depois vai iniciar a sua apresentação. Ok, Robert, could you please introduce yourself?
Okay, welcome everyone. It's my pleasure to be here and talk about 2D materials and safe design. I'd like to thank Dr. Martins for the invitation to do this. And as he said, I'm Robert Hurt, and I'm calling you or Skyping today from Brown University, which is in Providence, Rhode Island, on the east coast of the United States, not far from the city of Boston. And I've been here for 24 years now. I'm a chemical engineer by training, and I did my graduate work, my PhD work at MIT, which is just up the road. Since being at Brown, I was director of Brown's Nanosciences Institute for 7 years. And for 12 years, I was an editor of the journal Carbon, which is a materials science and nanotechnology journal focused on carbon-based materials. And for the last 5 years of that, I was the editor-in-chief. I rotated out of that position just last January, and now I'm full-time back at Brown, and I'm the director of Brown's Superfund Center, which is an NIH, or National Institutes of Health, supported center that focuses on human disease caused by environmental factors. So it's the area of environmental health, which is the topic we're talking about today is a subset of environmental health research. And I'm a chemical engineer, but I've always been interested in how materials behave in chemical and biochemical environments. And my research today in 2018 is about 2D materials, and both their applications, how to make them and what they can be used for to advance technology, and also their implications for human health and the environment. How can we ensure that they're developed and used safely and don't cause human diseases or environmental issues at the same time we're using them to solve societal problems? And this last part, the implications, is the topic for today under the guidance of Dr. Martins. So I'd like to talk about 2D materials and their design for safety. So I have a few slides I would like to pull up to guide the main part of the presentation. One second, let me share here.
Okay.
Can you see the slides?
Not yet.
Not yet? Okay.
Yes.
Yes. Okay. Good. So the safe design of two-dimensional materials. So, you know, this field really began, as I think many of the listeners know, with the discovery and isolation of graphene in 2004, which led to the Nobel Prize in 2010. And I've been working on carbon materials for decades before the discovery of graphene. But for all of that time, we had graphene in our minds, and not as an isolated layer, but as a building block for all carbon materials. And the name was defined way back in 1986 as the hypothetical end member of the series of polyaromatic hydrocarbons, from benzene to naphthalene to phenanthrene, all the way up to graphene, ene meaning the carbon-carbon double bond, and graph is from the Greek word to write. And I've worked on carbon materials for many years. It's both a new and an old material. And I like to show this next slide from a 30,000-year-old charcoal drawing from the south of France in a place called the Chauvet Cave. And this was done with charcoal from campfires or forest fires. And two things strike me about this image. First is just the beauty of this painting from the ancient Europeans, to be able to produce a piece of art so beautifully using something as simple as charcoal from a fire. And then the second thing about this is the similarity between this work 30,000 years ago and the 2004 work that led to the Nobel Prize. So if you scrape charcoal over rock, you essentially exfoliate that charcoal and produce what nowadays we might call multi-layer graphene deposits. And so we didn't have journals back then, but I imagined as an editor what the abstract of such a work might look like if the ancient Europeans had submitted it. And you could say that here we report that organic matter in the form of wood can be transformed by heating into solid elemental carbon and then mechanically exfoliated into highly stable, optically absorbing thin films that we propose to call multi-layer graphene. That's how old carbon is as a material. And since the discovery of graphene, it has motivated people all over the world to look out in nature for other naturally layered crystals and to find those that can be exfoliated and separated down to single layer or multi-layer packets. And you probably know this is developed into quite a large field in material science. Many things in nature are naturally layered with strong bonds in plane, but only van der Waals bonding between planes. And all of the minerals that have that structure can, in principle, be pulled apart by one method or another to produce atomically thin or near atomically thin two-dimensional materials. So today, this is 2018, and these 2D materials are best thought of as a broad class of high aspect ratios of length to thickness, either plate-like or sheet-like solids. They may be monolayers, they may be few-layer nanosheets, or they may be somewhat thicker, what people call nanoplates or nanoplatelets, packages of layers that are thick enough to be stiff, but still have extreme aspect ratios. They are much wider in 2 dimensions, much, much wider than they are thick in the 3rd dimension. And the general classes to make these materials are chemical and mechanical. In the mechanical method, somehow force is used, whether it's sonication or intense stirring or the Scotch tape method to rip apart layered materials into individual nanosheets. In chemical methods, intercalate some agent in between the layers to pre-expand the material, and then typically by heating or by solvent contact to decompose the intercalant, to have it come apart, create internal pressure, and blow apart, separate the layers of a layered material into 2D pieces. 2D materials can also be grown by chemical vapor deposition, the way we often grow carbon nanotubes, say, bottom-up atomic scale growth process, or they can be grown colloidally or solvothermally. It's quite a large field. And this field has now produced a large set of different materials of widely different chemistries. And we tried to review this field just 2 years ago in the Chemical Society of Review's journal. There's the reference given. And what we have now is a broad family of new materials. It's enormous chemical diversity, but also diversity in form, because the thicknesses can go from a single monolayer, so atomically thin, up to few layers into many layers, eventually to bulk materials. And along the other axis, the lateral dimension of these materials can be tiny, can be nanoscale itself, and those are often called quantum dots or 2-dimensional quantum dots, up to microscale materials, which are often called nanosheets with micron scale dimension, and all the way up to, it's difficult to make very large 2D materials with large lateral dimensions. People are trying, and there's some progress, but it's difficult. But this is a family of materials, and if you're interested in safe design, you can ask the question, how do we go about addressing such a diverse class of different chemical substances that all are sheet-like or plate-like, and do it in a way that ensures that human health is protected while these technologies are being developed? It's not an easy question, and my interest has been in making this process work better and finding new ways to address the whole family of materials. We can't start on this list and one by one go through each individual material and do elaborate toxicity testing involving animals. Other methods have to be developed. And so we have, in our review article, we talked about how this might be done. And the program of research that I've been interested in here at Brown is to look at a more deeper level at 2D materials, and instead of doing animal studies, find ways to understand the basic pathways that lead from that material to a toxic response. And if we can do this at the fundamental level, using chemistry and physics, and understanding what biological pathways are initiated we could hopefully maybe classify these materials into those that are maybe of special concern and worthy of more detailed studies of their toxicity and their safe design, and maybe other materials that we could expect to be less important, less likely to be hazardous, and maybe to act in many ways like conventional nanoparticles made from relatively benign constituents. And so if we can do that, we've made great progress in working through this family of materials to ensure that they're all safe without having to mindlessly go through them one by one with elaborate and expensive techniques. So we thought, what if we begin to look at 2D materials, as we see on this slide, and we look at the classes of their interactions with biological systems. We have chemical interactions of various types. We have electronic and surface redox interactions. We have physical and mechanical interactions. And in my little presentation today, and I'd like to leave time for questions at the end, I'd like to say a few words about each of these different types of interactions and what kind of work we can do to make 2D materials safe. And the first type of a way that a 2D material can interact in the human body is by creating free radicals. After, say, inhalation or implantation, chemical reactions often involving oxygen or hydrogen peroxide at the surfaces of particles can often produce highly reactive species, oxygen-containing free radicals that attack tissue and lead to inflammation. This has been known for many decades and is a major paradigm, a major way of thinking about particle toxicology that dates back to work on air pollution. So atmospheric air pollution produces fine particles. Some fraction of those we know enter the lung, and depending on their surface states, those particles can lead to free radical production that causes inflammation in the lung and can lead to eventually to lung conditions and diseases. This is a very kind of what classical way of thinking about particle toxicology, and it's the same for 2D materials as it is for other materials. It's potentially very important. However, it's not something that's particularly, it's not something that's unique to a 2D material. It's something that we've known about for a long time, and work has to be done to understand if which of these 2D materials, many of which are semiconductors, would in fact be redox active and produce free radicals in the human body. So I don't want to spend too much time on this mechanism, but go on to something that we're working on quite actively at Brown, which is understanding how 2D materials interact chemically in the human body. And so when you put materials, say you into the lung or into systemic circulation, into the bloodstream or even the GI tract, in the early days of nanotox, we thought they would largely be inert and they would just be particles, and that was the only concern, was what the particles would do. But now we know, after decades of research, that many materials transform. They degrade, they dissolve, they become oxidized, they become reduced, they transform into other phases, and when they do that, they become new materials, and now we have to be concerned about whether this new material or the soluble dissolution products may in fact be causing a biological response. Our focus has to shift from the particle itself to the transformation products, whatever they are. So understanding how a material transforms in the body is really very basic to understanding its risk. And of course, so particles influence biological structures, but as a material scientist, I'm interested in the opposite of that. How does the biological environment, how do organisms and tissues, how do they influence materials and change them? That's one of the topics that's a particular specialty of our laboratory. So I have a few examples of work in this area of chemical interactions for 2D materials. And the first one, let me talk a little bit about these interactions. If you go back before 2D materials and look at the general nanotoxicology literature, we've learned a lot. The things that nanoparticles can do if they enter the natural world, if they enter the body. They can dissolve or degrade, change phase. They can get modified on their surfaces. They can absorb things from the body and change their surface chemistry. They can become coated by macromolecules like the protein corona. They can agglomerate, they can aggregate. Probably the most important transformation is degradation. If a material degrades, it is not persistent in the human body. And many of the most hazardous particles that we know, asbestos fibers, and say silica, crystalline silica, are hazardous because they are persistent. Asbestos can remain in the body for decades, and things that remain intact as solids in the body have the chance to produce inflammation, chronic inflammation over long periods of time that can lead eventually to the development of cancer. Materials instead that degrade, they may be acutely toxic and there may be short-term effects, but if they degrade and the degradation products are cleared, those materials tend to not present longer-term health hazards in the same way that persistent materials do. So I think the most important thing we'd like to know about all of these 2D materials as we start is which ones will be stable in the body, which ones will stay around as particles over years and potentially cause effects, and which ones will go away. And now, of course, the makers of 2D materials they're being designed and made for very different purposes. Most are not being made for applications in the body. So we believe that most of these materials, their stability in human body fluids is unknown. And they're often studied in vacuum, they're studied in dry air. They're not typically diluted into salt solutions like we have in the body, and they're not exposed to oxidants and reductants in the highly dilute phase. So we did in our review article a summary that made the prediction that many of these 2D materials would in fact not be stable under the conditions relevant to human health. Their developers claim they're stable under their application conditions, but under the conditions we're interested in our field, nanotoxicology, we think many will not be stable. And I have a case study to start with, and this is a material produced by a collaborator of ours, Christy Koski, who's now at the University of California, Davis. And these are molybdenum trioxide nanoribbons that she makes in her lab. And they're 2D materials because they have van der Waals gaps. They're stacked sheets of MOO3, but they also tend to be long, so they're a sort of hybrid 2D, 1D nanomaterial. And if you look at them, they look very stiff and sharp and pointy and long, elongated. They have many things in common with the geometry of asbestos fibers. And so a person familiar with the human response to asbestos exposure would look at these materials with some natural concern. And the first thing we asked Christy was whether these materials were stable in water. And she says, yes, of course, because she makes them in water by a colloidal method. So these materials appear to be stable to the people making them, and they have asbestos-like geometry. So we thought we would study these, beginning with the issue of persistence. But what we found out very quickly, actually, was that these materials, when they're diluted into the kinds of concentrations that one finds, say, in the human lung, or under injection into the bloodstream, or in the GI tract, they seem to dissolve quickly, especially in buffered solutions with controlled pH. And you can see the material on the images going clear after 20 minutes, these white cloudy suspension with these white needle-like molybdenum trioxide nanoribbons goes clear, which suggests to us dissolution in a pretty short time. It doesn't happen in simple water solutions, but it happens in, say, some cell culture media, and it happens in buffers. And the data on the right show that if you measure the amount of dissolved molybdenum in the course of an hour in many media, they disappear and dissolve. And we eventually found out that molybdenum was undergoing a basic hydrolytic dissolution process to produce molybdate, which is a soluble ion, and gives off proteins. And in simple water, protons. And in simple water solutions, those protons make the solution acidic, and the molybdenum reaches an equilibrium. So we have a stock solution with nanoribbons and some dissolved material. But if you look at conditions found in the body, conditions where the particles are ultra dilute and where there's buffers that maintain the pH at levels needed, say, for cell growth. Under those conditions, these materials are very unstable. And while they have asbestos-like geometry, we also find in cell work done by Cynthia Browning, who's shown on the slide, in cell work, they dissolve and degrade before cells ever have a chance to see or respond to their asbestos-like shapes. And these materials, based on persistence ideas alone, we would classify as a relatively low hazard for human health. And it's a kind of material that we think can be safely developed as we move forward for applications. And the added benefit of this material is that when molybdenum trioxide dissolves into soluble products, those soluble products, molybdate ion, is not particularly toxic in its own right, so the shape goes away and the soluble products are relatively safe under most doses, making this material something that we think could be safely developed. Another good case study is molybdenum disulfide, and this has widely been reported in the literature to be very stable. Molybdenum disulfide is used as a lubricant now, as a catalyst. It's recently been exfoliated into single layers and, for electronic applications. It's highly insoluble. If you put molybdenum disulfide in water, its solubility is extremely low. But we've found, and then others have also been finding, that it's not fully stable over time in the presence of oxygen. So living things need oxygen to metabolize, as oxygen's ubiquitous in the human body, and molybdenum disulfide, if put in biological simulant fluids, undergoes a slow dissolution. And this graph shows that over, now this is 100 days, but over 100 days, this material will slowly degrade, and the molybdenum will appear in solution as a dissolved product. The nanosheets will be destroyed. If you remove the oxygen by bubbling, say, with nitrogen or argon, if you take the oxygen away from the system artificially, which you wouldn't do in the body, it can be done in the laboratory, these materials retain their stability. So this is an example of a highly insoluble material, but it has an oxidative instability that most researchers wouldn't see unless people studying the transformations of these materials in the human body take a special look under our conditions relevant to human exposures. So we believe molybdenum disulfide, although thought to be quite stable, will in fact be also in a class of materials that does not have long-term persistence in the body. Now, because 100 days is relatively long, if you breathe in molybdenum disulfide, it will exist as a nanosheet for some time. Macrophages will clear it, hopefully from the lung, but it will persist for potentially some months within those cells, possibly be swallowed as the macrophages move up the mucociliary escalator and it becomes swallowed or otherwise removed. But in terms of long-term persistence and long latency disease formation, such as we see with asbestos, we believe this material also is not stable enough, and it's an important finding. Now our final, and this is the chemical reaction equation. It shows molybdenum disulfide reacting with oxygen, again to produce protons. So in buffered solutions and with patients, one can see its degradation. And here's a reference to the paper 2 years ago that discusses this. We have a third type of case study, which I can't talk about today because we have unpublished data, but some 2D materials are stable in water. They're stable to oxygen, but they are unstable in the presence of biological reducing agents. So the body has antioxidants, glutathione, cysteine, ascorbic acid, a variety of compounds known to neutralize free radicals. Those are chemical reducing agents. And when these materials see those chemical reducing agents, which they don't normally do in their applications or their handling, but after exposure to human beings, they will, there's another class of materials that become unstable and dissolve under reductive conditions. And so this has led us to sort of develop and propose this framework, which is ends this middle part of my presentation. And this framework is our suggestion for how one could go about looking at the whole set of 2D materials and working through them to understand which ones may be hazardous and which ones may be more safe for development without a large body of additional work. And this classwork, this framework starts at the top. And if we have a new material, we would suggest that one does a literature search on not on the 2D material alone, but also on its bulk form, the bulk layered material, many of which are found in nature, and understand what its dissolution pathways are. Is this a material that can be oxidized? Is it a material that can be reduced? Or is it a material that might dissolve on its own under dilute conditions, either in acids or bases? And based on that, one can do simple tests, dissolution kinetic studies, in a simulant that is picked to match the dissolution pathway that you would predict. If it's oxidative, you do work on air-saturated solutions. If it's reductive, you'd add an antioxidant like glutathione. If it's a non-redox, let's say, dissolution that's favored by acid, then you might study a simulant that simulates what happens inside macrophage cells in their lysosomes, which have low pH, like 4, which can favor the dissolution of some materials. And these are very simple studies compared to the difficult and expensive animal studies we normally do. But as a result of those, we think you could classify these 2D materials into 4 groups. There's a Class A group that's where we see no significant dissolution. These are potentially biopersistent materials, and they would be at the highest priority for full characterization of the solid phase. And we would treat these the way we treat most nanoparticles, understand their shapes and sizes and surface properties, and expect the particle to remain for a long time and understand what that particle can do in the human body. On the other end, there's materials that undergo rapid dissolution. And here, the framework splits into 2 classes, because if something dissolves rapidly, say within a few days, you may no longer care so much about the shape and the properties of the particle, because the particle or the sheet doesn't last. But you would be interested in the properties of the dissolution products, and sometimes they're hazardous, and sometimes they're less hazardous. So a material that dissolves rapidly into soluble materials that have a low hazard, and this is a chemical hazard that you can understand from existing literature in most cases, or it can be measured separately. Those would be Class D materials that would be of the lowest priority for further testing. And our molybdenum trioxide nanoribbons would be an example of a Class D material. Dissolves quickly into soluble products that have a low hazard. And those can be set aside, and that helps us move through the family of 2D materials in a rational way. Then there's materials that dissolve quickly into hazardous products. And an example of that might be nanocopper, which often oxidizes and dissolves in the human body, but produces copper ion, which is redox active and can cause inflammation. So these are potentially hazardous materials, but we can probably understand their hazard better in terms of the chemical hazard associated with any copper form. And we might not need detailed studies on the particles themselves, which are just sources of the chemical, in this case, copper. And then finally, sort of in the middle is a more complicated Class B of materials that dissolve but slowly. And an example of this would be molybdenum disulfide. These would be likely non-persistent, so that reduces the concern about their effects, say, relative to particles like asbestos or crystalline silica. But they do last for a while, and one is exposed both to the solid and to the particles and to the ions or the soluble matter at the same time. So we think these are worthy of future study, but one might want to focus on acute short-term effects rather than long-term effects, because these materials will not be present in the body in this form for a long time. So this is the framework we've been proposing. It's about to be published here in the next few weeks, and some of the examples of members of these different 4 classes would be shown here in red. An example of a potentially biopersistent material would be the thick multi-layer graphene or graphene nanoplate materials that are being produced by many companies. These are little sheets of graphite, essentially, And we're not saying they're hazardous, but they're quite stable. And we could imagine they may exist in the body for a long time, and future work would need to be done to understand their safety. Nanocopper, we expect to dissolve into copper forms, more traditional type. Molybdenum trioxide nanoribbons are the class D material, and molybdenum sulfide, intermediate case. Now, those are an example of the chemical interactions, but let's say you have a material like a multi-layer graphene material, which is expected to be stable. Well, we need to look at that material carefully in the solid phase. And that leads to the last set of interactions I'd like to talk about, which are physical and mechanical interactions. And this is probably the most important topic in 2D materials because they represent a diverse range of chemistries, but what they all have in common is the sheet-like geometry. If you say you're working on 2D materials in biology, really the fundamental question is how does an ultra-thin nanosheet, which is quite different than the things we find in nature, typically, an atomically thin nanosheet, how does it interact with biological structures through its very unusual shape? That's a fundamental question, and we know right now far too little about how nature perceives atomically thin, sheet-like, but yet stiff and sometimes rigid materials. That's a kind of an unknown, and it's something that the field needs to look at very carefully, and it's a problem that's common to all 2D materials. And we've done a little bit of work on this. The next slide just shows some examples of things that atomically thin sheets might do when they encounter cells. The center diagram is a kind of a directory to the different images, and you can see these yellow nanosheets, all the things you can imagine, and some people have seen that they do, that could potentially cut the membrane. Being so thin, like a knife, they can spontaneously diffuse across a cell membrane because the energy required to part the lipid phase is so small. Some people have suggested they can reside inside the cell membrane in the hydrophobic core, as you'll see in B. They may have things absorbed to them, as in A, and they may cross the cell membrane and carry in components from the outside into the cell through passive diffusion. Being flexible, they may sit on the outside of the membrane, as you see in F and G. They may be taken up inside the cell, and inside the cell they may be packaged in lysosomes. Cells try to package away foreign objects into spherical lysosomes, and it might work for a nanosheet, but for nanosheets that are large, is it possible for the body, for the cell, to package them away in a sphere if they're too large to fit inside a lysosome? These are all important kinds of interactions that 2D sheets may have with cells if we knew more. And the field has started to work on this. We did some work just a few years ago trying to understand how graphene materials were taken up inside cells. And in this work, these are graphene microsheets, and we found them inside cells. This is my collaborator Agnes Kane in her lab. We find them inside cells. And it looks like if you try to capture the process while it's happening, as the sheet comes down, and because in the process of entering the cell, we often see the sheets standing up on end with their edges partially inside the cell and the rest of the sheet outside the cell. So it seems like they're entering edge on. And I have a modeling colleague, Professor Huajin Gao, who ran molecular dynamics simulations of this, and he would show that small graphene sheets can in fact spontaneously enter lipids this way. But if he looks at a large sheet, like the kind we're using experimentally, those sheets would not enter the lipid membrane easily. They would be repelled. It was difficult for a sheet of micron scale dimension to cut through a lipid bilayer. But what turns out to be the case is that the actual graphene sheets, if you look closely at this image on the left, they're very ragged. They don't have smooth edges. And if you bring an irregular edge of a graphene sheet up to a cell, it's no longer smooth, and the sharp corners can puncture the membrane, pass through the membrane, and in Huajin Gao's theory, he believes that that puncture event starts the process of entry and allows the sheet then to spontaneously pass through cells and enter them. And this is work that was published in 2013 in PNAS if you're interested. So now entering a cell is not necessarily bad. Macrophages try to gather up particles in the lung and remove them. So if they're able to take these particles up, that's not a bad thing. It can be a good thing. But the issue with 2D materials, we believe, is that ones that are small may be taken up inside cells like particles, as you see on the left. And as the sheets grow in size in lateral dimension, eventually they become too large for macrophage cells to clear. So it seems likely to us that very large graphene sheets or large lateral dimension sheets of any 2D material, if they're thin enough to enter the lung and get into the deep lung, but they're so large that single cells can't take them up and remove them, then they may stay in the lung for a prolonged period of time. Other macrophage cells are recruited, and one produces a granuloma or a tissue foreign body response inside the lung that may be a cause of longer term or chronic lung conditions. So currently we suspect that the small nanosheets act somewhat like particles, but larger nanosheets may challenge cells, may avoid macrophage uptake and clearance, and may be more of a problem in the human lung. But my last slide, so how do we address this? Well, my last slide is not about 2D materials at all, but it's about a 1D material, carbon nanotubes. And it's our most recent work, and it's a phase diagram that we just published also in PNAS last year. And what it does, it tries to define which kinds of nanotubes can be taken up in cells and packaged away successfully, and which kinds lead to adverse biological responses. And we found that the carbon nanotubes longer than about 1 micron begin to cause problems in cells. And what this phase diagram shows, it's a complicated diagram, but if you look, we're plotting the length of the nanotube on one axis and the diameter on another, and this big thick blue line is a buckling threshold. So nanotubes that fall to the lower right of that line are stiff. They can't be buckled by biological forces. And nanotubes that fall above and to the left of that line are soft, flexible, more like noodles than like needles. And these noodle-like nanotubes don't appear to the cells as fibers. And in our data, as cells try to take up nanotubes and package them in lysosomes, the ones that are long enough and stiff, lying to the right of that blue line, end up causing pathological responses as the cell cannot properly package that stiff fiber into a lysosome. The other types of nanotubes can bend and curl and be packaged in lysosomes as soft structures, not hard structures. And I would like to leave the group today with just an idea that for the field, can we find such a phase diagram for 2D materials? Exactly what thickness, what stiffness, and what size of a 2D sheet is really important for determining if those materials would be safe when you breathe them into the lung. If we can understand that basic question, we could apply it to many different 2D materials currently being developed, and we would have made much progress in understanding fundamentally how to design a 2D material for safety. That's something the field really should look at in the next few years. So thank you for listening, and I'm happy to take questions from Dr. Martins.
Yes.
If he would like to ask some.
I don't have enough knowledge to put some in deep question. Okay? So I will put some general question. Okay? One is about the costs. How much costs this kind of research?
The costs?
Yeah.
It's a good question. I think biological safety studies, the cost, traditional tox testing used for regulatory regulations, involves typically cancer as an endpoint, animal studies, often 2 species and 3 doses, and millions of dollars to do a study on a single material. So it's not very practical, and people have begun to realize that we can't go forward here in 2018 studying every chemical or material that people have made that way. And the European Union, with its REACH regulations, is pushing very hard to move beyond animal testing and find other more science-based, laboratory-based, cell culture or beaker-based tests to predict if things would be hazardous or toxic, to allow us to go through these questions without spending a fortune of the government's money. And so the work I talked about is part of that effort. And some of our experiments are embarrassingly simple. You know, these biological dissolution experiments are something that many chemistry labs can do. You need a way to assay for the soluble material. You need, it's often metal-based using, say, ICP or other techniques. These are chemistry experiments done with biological molecules, and those are not, in the world of research, those are not expensive types of studies.
Okay, the second question is about the bottleneck. What are the bottleneck to develop more this kind of research?
Yeah, that's a very good question. I think obviously there's been quite a bit of research on nanotoxicology. It was a very hot topic for at least 10 years, and a fair amount of government funding was put into it. There's still continued funding for this area, but not as much as there was for a period of time. One of the bottlenecks is that small companies are often developing these materials, and they don't have the departments and the staff to do this kind of work. Some small companies have a handful of employees, and they're all interested in the product, and they don't have an environmental health specialist or someone who can do anything to find out how hazardous their materials might be. So larger companies are not as challenged. They have groups that do this, and they understand very well how to produce products and sell them that don't hurt people. But smaller companies that are driving the innovation in these areas often are not equipped, personnel-wise or equipment-wise, to do anything about the potential safety of their materials, and they need to reach out and have resources that can work with them. That I see as a bottleneck.
Okay, I would like to tell you thanks so much for your contribution to our program, and when your paper will be published, after this you can do another program we'll talk about this.
Okay.
Okay.
Thank you.
Thank you so much.
Thank you, Dr. Martins. Thank you.
Thank you. Bye-bye.
Bye.